Can somebody confirm you can hear me?
I can hear you.
Thank you, Kelsey. All right, before I share my screen, let me talk a little bit about the grading. I guess before we start doing anything, are there any questions? I want to show the grade book because it'll help me talk about the grading. And obviously, I don't want to share my screen when I'm looking at the grade book. So I got the environmental quiz graded. I think I got that one graded a long time ago. The practice quiz 00B is graded. However, there's a bug in the Canvas. To see the grade, you gotta click on the icon, which actually looks like it's something submitted for submission and it's not graded. But if you click on it, it'll show you the grade. And then Plagia Quiz 1 is graded. Hopefully people got 19 out of 19. The extra credit picture I've not graded yet, but the extra credit study sheet I have graded. I need to grade extra credit study 2 and 3. Sorry about that. I do have worksheet for lecture 1 and for lecture 2 graded. Usually in the worksheets for the lecture, you're just answering 1 or 2 questions. You must answer the questions in the order they are in the worksheet to get full credit, and you must answer a question that somebody has not already answered. So you should take a look at your answer after you posted it to make sure somebody did not submit an answer before you posted yours, because the first person will get credit, the second person and anybody later will not get full credit. So to get full credit, you'll need to post another answer, meaning a question that has not been answered. Also, if you do not know the answer to the worksheet question in the lecture, then you should look it up because this assignment will only be graded one time. I do have the worksheet for lecture 3 graded as well. Let me move 4 because it's out of place. It should be after 3. There we go.
Are you sharing your screen?
No, I'm not sharing my screen because I'm looking at my grade book to explain the grades, what I've got
Okay.
graded. And I won't remember this, so I've got to look at the grade book. So quiz 1C is graded. And I believe I gave everybody 6 points or 2 questions of credit. So I threw out 2 questions and you got 6 points bonus. That actually made 2 people 100% or higher in the grades. And that helped some of you. Obviously, you got a really poor grade. It wasn't enough to help you, but it helped you a little. So take a look at that. And I have the lab 0001, lab 2, lab 3, lab 4, and lab 5 all graded one time. Lab 00 and lab 1 were only going to be graded one time. I need to go back and grade lab 2 and lab 3 a second time. And then lab 4 and lab 5, you have until this Saturday to submit your answers if you didn't get 100%. So if you didn't get 100% on lab 4 and lab 5, you can resubmit the lab by this Saturday. I think you have until 11:59 p.m. this Saturday to submit lab 4 and 5. Any question about any of that? On the first grading, I'll tell you what questions you got wrong. I may give you a hint, I may not, depending on what the question is. Like those of you who got partial credit on Lab 5 for question 9, I think I gave you a hint saying that you submitted one plate, and there are actually two that are not strict for colony isolation. But I don't always give you a hint. I may just say something like question 8, the bat plate was partially wrong or completely wrong. Any questions about any of that?
It's my understanding, I know that this is not your policy, but just wondering what happened along the way. But it used to be that in microbiology, it's my understanding from other students, it was open notes for quizzes. And like, was there a reason that changed?
Yes, I made it closed notes to reduce cheating, and also there are a few other reasons I don't want to get into why I made it closed notes. I can email you specifically if you send the question there, but um, I recently began using Respondus Monitor, and I thought I was giving the students enough time to look up 2 or 3 questions, and it turned out that most of the students, about half the students, were actually looking up half the questions on the quizzes. And then for the special needs students, some of them were looking up all of the questions on the quizzes. And I decided I intended it only for 2 or 3 questions to be looked up, and many more than 2 or 3 questions were being looked up. So the exam is now much more similar to if you took the quiz or exam in a in-class exam where it's closed notes. And yes, that's my policy. Why did that end? Don't know. All right. Any other questions?
I had a quick question about the first quiz that we took. Well, I mean last week's on Wednesday. You had mentioned that you took two questions out and gave 6 points. Is it because the two questions were like I guess across all students they're getting it wrong, or I was just curious.
No, not all students got 2 questions 100% wrong.
Okay, sounds good.
Okay.
Well, I'm not mad.
All right.
Okay, sweet. Thank you.
All right. All right. Let me get out of the grade book and then I'll share my screen.
I had a quick question. Do you have
Uh-huh.
any accommodations for students that do or might need them for the testing using the Respondus app?
Meaning you're having trouble with Respondus Monitor?
No, like the school's accommodation for students that might need like you said earlier you said special needs students were doing stuff. So I was just wondering like what accommodations for them with the program. I can't find that in the syllabus.
Um, if there is a student who gets an accommodation, generally I give them the accommodation. So it would have to be an accommodation that I don't remember what the department is called now. It used to be the Disability Department or Department of Disabilities. I don't remember what it's called anymore, but I have to get the accommodation in a letter from them, and generally I give the student that accommodation. Okay?
Perfect. Thank you.
All right. Oops, that's the wrong one. Let's go here. All right, so today is April 29th, and we're going to talk about Lab 6, Juice Diffusion and Antibiotics. You do need to read the Unknown Project instruction file on Canvas for next week. I'm going to talk about the Unknown Project, and geez, we're going to be starting it next week. I'm not quite ready yet, but I got another test to make too. Well, hopefully I can get everything done. Let me see. I'm going to try and show you where the Unknown Project file is. Scroll down to week 5 lab. Whoops. There we go. So week 5 lab, the unknown report, the unknown instructions. This is the file I want you to read because I'm going to talk about it next week. And it'll really help if you have read it so you know what the heck I'm talking about because I'm not going to talk about everything in the file. All right. Any questions about what we're doing? Remember, we have a quiz tomorrow, and I have to make part of that quiz. That's why I've got a lot to do by tomorrow. And I got to do it tonight because the quiz opens at midnight tonight. But the quiz will be covering the end of chapter 2, chapter 3, and then labs 3 and 4.
I have a quick question. Is there like what end of chapter 2, just so in the PowerPoint, are you able
It's
to say?
about, we had spent 2 days on chapter 2, so the first day will not be covered on chapter 2.
The first day.
It's someplace near talking about organic molecules, but I don't remember exactly where we left off. You can take a look at the recording online if you really need to find where the division is.
Okay. Thought I tried, but I didn't. I'll look again.
Okay. Sorry, I just don't remember off the top of my head where we began, where we ended, but it'll be the first day.
I
It's
think
not going to be covered.
I think it was stated in last Monday's video. I think someone had asked at the before the test, and you had said that last Monday's would not be in Wednesday's test. That's just off the top of my head. But
The first day for chapter 2 will not be on this coming quiz. The second day for chapter 2 will be on this coming quiz.
okay, sounds good. Okay, thank you.
Okay. All right, I'm having trouble. There it is. Hopefully it's lab 6. Oop, not quick enough. All right. So, lab 6, diffusion, evaluation of disinfectants and antiseptics, and antibiotic effectiveness. You need to do some reading in the textbook before you do the lab. You should read about the terminology of microbiological control. One page, principles of effective disinfection, evaluation of a disinfectant, and types. A couple of pages, page 187 to page 194. You need to read about chemical methods for microbial control, about a page. And you need to read about antibacterial drugs or antibiotics, and that'll be about a page. And then the tests to guide chemotherapy, the disc diffusion method, and that's page 567 to 569, or about 2 pages. The disc diffusion method is sometimes called the disc diffusion method, the Kirby-Bauer method, and evaluation of antibiotic effectiveness. So all of those terms mean the same thing. There are supplemental materials in this lab. There are video clips and a virtual lab for this lab. The learning objectives: 1, be able to identify the chemical agent and the active ingredient used in various disinfectants and/or antiseptics. 2, evaluate the effectiveness of disinfectants or antiseptics using the disc diffusion method. And that's also the same as using the Kirby-Bauer method. 3, determine the mode of action of various antibiotics. 4, evaluate the effectiveness of various antibiotics using the disc diffusion method. And number 4 is what this lab is mostly going to be looking at. 5, list the variables that influence antibiotic disc diffusion susceptibility testing, or the Kirby-Bauer method. And 6, define the terms. What's sterilization, disinfection? What is a disinfectant? What is an antiseptic? What is a bactericidal agent? What is a bacteriostatic agent? What is an antibiotic? What's the spread plate method? And what is a zone of inhibition? Any question about any of that? All right, you should know the terms. Sterilization refers to the elimination of all viable organisms, including endospores. This may not mean prions or viruses, but mainly prions. Because sterilization doesn't really work with prions, and viruses really aren't alive either. But it does remove endospores. And how we sterilize things in the normal microbiology lab is we stick them in an autoclave, which is a giant pressure cooker. And I think we'll talk about that in this lab if we haven't talked about it already. A disinfection refers to the destruction of vegetative cells, but not necessarily endospores. So when you disinfect your toilet, you may not be killing endospores, but you should be killing all vegetative cells. A disinfectant is a chemical agent applied to an inanimate object. An antiseptic is a chemical agent applied to a living tissues. So, if I were to clean my toilet with bleach, I would be using a disinfectant to clean the toilet. But if my cat, I don't have a cat by the way, were to come in bleeding profusely and I would clean the wound with bleach, because that would be the only agent I had, let's say, I would be using an antiseptic on the cat. So in this case, bleach has been used for two different purposes. And it's the same agent, and once it's an antiseptic, the other it's a disinfectant. It used to be with necrotizing fasciitis, the flesh-eating bacteria, that they used to treat it by putting one treatment, I guess I should say, because they had more than one. They usually give the patient antibiotics and then treat the tissue that's infected with RAP that is inside bleach. I don't know if they still use that, but it used to be a treatment for treating necrotizing fasciitis. Any question about any of that? All right. To evaluate the effectiveness of chemical agent, the surface of a nutrient agar plate is inoculated with a particular bacterium. Paper discs, then saturated with chemical agents, are applied to the surface of the agar. The plate is then incubated and the chemical agent diffuses into the agar medium, establishing a concentration gradient around the disc. After overnight incubation, the bacterial growth pattern around the disc is observed. If a zone of inhibition is seen, meaning an area with no bacterial growth around the disc, the chemical is assumed to be effective for that particular bacterium. Doesn't mean that the bacterium is sensitive to that chemical agent, but the chemical agent is effective. Conversely, if there's no zone of inhibition, the chemical agent is assumed to be ineffective. Any question about any of that? So this table is showing you the common chemical agents like ethanol, used preferably as an antiseptic, but it's a very weak antiseptic, so we call it a degerming agent, meaning when we use ethanol or isopropanol on the skin, we don't remove all of the bacteria from our skin, so we call it a degerming agent or a very weak antiseptic, but it makes the area safe so that when you stab the needle through the skin and give an injection, that there be very few bacteria injected under the skin, so the patient is likely to not have a problem. Halogens like iodine and chloride. Iodine is usually used as an antiseptic, and then chlorine bleach is usually used as a disinfectant. Any question about any of that? You can take a look at the table. The zone inhibition size you get depends only partially upon the sensitivity of the organism to the disinfectant or the antiseptic. There are various other factors that affect the zone size, including the concentration of the chemical. Usually the more concentrated the chemical, the larger the zone of inhibition. The molecular weight of the agent also affects the zone size. Generally, the smaller molecular weight of the agent, the larger the zone of inhibition. And then the density of the bacterium inoculated. The denser the bacteria that's inoculated, the smaller the zone of inhibition. So when we're looking at the zone size, certain assumptions have to be made, and we standardize the procedure to control many of these variables. Evaluation of antibiotic effectiveness. Antibiotics are categorized as bactericidal if they kill the bacteria, or bacteriostatic if they inhibit bacterial growth. Any question about that? Do you all see the distinction? So, if you're treating with a bacteriostatic agent, it does not kill the bacteria. And then once you stop treating with the agent, the bacteria may begin growing. If you're treating with a bactericidal agent, it will kill the bacteria. When bacteriostatic agents are used, we are relying on the immune system to assist in the elimination of an infection. Any question about any of that? Penicillin G, for example, is bactericidal. I don't know one off the top of my head which is bacteriostatic, but I think I've got one in the lecture that I'll talk about later. Let me see if I went too far. Nope. All right. The Kirby-Bayer disk diffusion method, also called simply the disk diffusion method or the method to evaluate for antibiotic resistance, and we'll call it the Kirby-Bayer disk diffusion method in this lab from here on, is a method for testing for antibiotic resistance, and it's the most common method used. The results of such testing help physicians in choosing which antibiotics to use when treating a patient with a bacterial infection. The bacteria are isolated, grown on an agar plate. The filter discs are impregnated with an antibiotic applied to the surface of the agar, and then the agar plate is allowed to grow, and then the plate is looked at, and we look for a zone of inhibition around the disc. If the antibiotic concentration is high enough to inhibit bacterial growth, we will get a zone of inhibition. Let me blow this up. The inhibition zone size is dependent on several factors, like the diffusion rate of the antibiotic. A small molecular weight antibiotic will diffuse further than a large molecular weight antibiotic. And so the zone size there could be just due to the molecular weight difference of the antibiotics. The zone size is also dependent on the sensitivity of the microorganism. A bacteria that is more sensitive to that antibiotic will have a greater zone size than a bacteria that is not sensitive to the antibiotic. And I don't have another antibiotic to show you. I mean, another bacteria to show you for that antibiotic. And then the growth rate of the bacterium will also determine the bacteria zone size. Let me see if I can remember this. The quicker the bacteria grow, the smaller the quicker the bacteria grow, I'm getting confused here. The slower the bacteria grow, the smaller the zone of inhibition. So the quicker the bacteria grow, the larger the zone of inhibition. Anyways, there's many factors that can affect the zone size, called the zone of inhibition. And so we use a standardized procedure to control these variables when performing the Kirby-Bayer diffusion test, or the diffusion method. And here's the standard protocol for the Kirby-Bayer procedure. First, you inoculate nutrient broth using several bacterial colonies grown on non-selective media, grown for no more than 24 hours old. You then grow the media in the broth to get turbidity of the broth of 0.5 Macfarlane units. That's approximately 1 times 10 to the 5th cells per mil. You then inoculate this broth at room temperature onto Mueller-Hinton agar plates. The different media can affect the zone size of inhibition. So we always run the Kirby-Bayer plate method on the Mueller-Hinton plates. And the agar is always between pH 7.2 and 7.4. I don't know how the agar pH affects the zone size, but probably if you have a pH at a different pH than this, then the zone of inhibition will probably be larger, but I'm not positive on that. And the depth size of the agar also affects the zone of inhibition. So we always use the Mueller-Hinton plates at 4 millimeters agar thick. And we always use 150 millimeters agar plates. If the agar is deeper than 4 millimeters thick, then the antibiotic will diffuse less and you'll have a smaller zone of inhibition. Any question about any of that? We always swab the bacteria using non-toxic swabs, using the sprayed plate technique to obtain confluent growth so the bacteria will grow everywhere on the disc. And in this lab, I'll go through that method in a little bit. You then place the disc at least 24 millimeters apart, meaning the paper discs are added with the antibiotics on them, and the paper discs are placed at least 24 millimeters apart, and no more than 12 discs per 150 millimeter plate. You add the antibiotic disc 5 minutes after you swab the bacteria onto the plate, but no longer than 15 minutes after the bacteria are added to the plates. You then allow the plate to sit for a few minutes, and after 15 minutes, you then incubate the plate in a non-CO2 incubator for 16 to 18 hours, and we always grow them for 16 to 18 hours at 35 degrees Celsius. And you always do that 15 minutes after you add the antibiotic disc. Any question about any of that? And we use all of this standard procedure so that you control the variables so that the zone size of inhibition will not differ for one specific bacteria, for one specific antibiotics, wherever you do this experiment in the world. All right, somebody had a question?
Yeah, I do. When it says like add the antibiotic discs 5 minutes, but no longer than 15, so you'll inoculate it and you have to wait at least 5 minutes, but you can't go past 15 minutes. Is that what I'm understanding?
Yeah, after you put the bacteria on the plate, you have to put the antibiotic disc on the plate 5 minutes later, up to 15 minutes later. The reason why you do 5 minutes is to allow the liquid, when you're adding the bacteria to the plate, to be absorbed into the agar. So that's why you wait 5 minutes. And then the bacteria can begin growing. So you always add the disc before or up to 15 minutes after you add the bacteria to the plates. So you
Okay,
really
and then
need
it
15 minutes.
and then it has to be incubated no longer or
15
like,
minutes later after you add the
okay.
paper disc. Yes.
Okay, thank you.
All right. And we do have a tool for adding the paper disc all at one time. So it's not a big deal. You just have the antibiotics on the tool, and then you put the plate under the tool, then push down, and it puts the antibiotic disc on the plate. That's why it's all uniform. And so just putting the antibiotic on the disc is not time consuming. You don't have to put that one down, then that one, then that one, then that one. You just put them all down at once. Takes only a few seconds. All right, after incubating the plates, after 16 to 18 hours at 35 degrees, you then look at the plates and look for the inhibition zones sizes. If there's a zone of inhibition, you then measure it along its diameter in millimeters. And the National Committee for Clinical Laboratory Studies sets a standard for interpreting the zone size where the bacteria is considered to be sensitive to that antibiotic, intermediate to that antibiotic, or resistant to that antibiotic. So here we have an antibiotic disc. There's the bacteria that's been plated on the plate. And there we have the zones of inhibition. You measure the diameter. Usually you take two measurements, one here and then one someplace else. Take the average. And this looks like 20 millimeters. And this is the antibiotic penicillin G. It just says penicillin, but assume it's penicillin G for Staphylococcus aureus. And then the table says, the NCCLS table says that if it's 20 millimeters or less, this bacteria is resistant to this antibiotic. So penicillin G here is resistant to Staphylococcus aureus is resistant to penicillin G. But if the zone were to be 21 to 28 millimeters, we'd say it's intermediate. And if the zone of inhibition were to be 29 millimeters or greater, we'd say that this bacteria is sensitive to this antibiotic. So the table will have each antibiotic and each species. And it'll have up to 3 different figures on it. One for the size of the zone of inhibition that's resistant to that antibiotic, one that's intermediate, that's in between, and then one that the bacteria is sensitive to that antibiotic. And so obviously there will be a lot of figures in the table because there will be a lot of different species, a lot of different antibiotics. And it's even a little more complicated than that because some of the tables have more than one concentration of antibiotic that they're looking at. But some of them only have one. Any question about any of that? So that's how we say whether a species is sensitive to a given antibiotic. You have to grow it out, measure the zone of inhibition size across its diameter in millimeters, and then look up that number in the table from the NCCLS table to get whether it's sensitive or resistant or intermediate. Any question about that? All right, you can know the terms of this lab. Don't need to look at the references. You're not going to be physically performing any of these activities, as everything we do will be done online, but you will be responsible for how to interpret the effectiveness of antibiotics using the Kirby-Bauer disk diffusion method. We have a little video for you to watch on preparing a spread plate, then review the steps for using aseptic technique when preparing a spread plate or a bacterial lawn of growth, meaning getting the bacteria to grow everywhere on the plate. And let me go through that right now. How we get the bacteria to grow on the plate. So pretend this is a plate. Hopefully you can see this. Put it in front of my face. Usually you can. If you can't, let me know. And then you get your loop. Pretend this is a loop. Put not the loop. It is a Q-tip. Put the Q-tip in your bacteria stock culture. This is obviously much bigger than it should be, but when you get the Q-tip wet from your solution, you then touch it on the edge of the tube to get the worst of the water off. And then you spread the Q-tip everywhere on the plate, which I'm not quite doing everywhere, but you're getting the idea. And then you rotate the Q-tip a quarter of the turn, rotate the plate a quarter of the turn. And then you do it another time, going everywhere on the plate. And then when you're done, rotate the Q-tip a quarter of a turn, rotate the plate a quarter of a turn, and then do it a third time, going everywhere on the plate. So we're swabbing everywhere on the plate to get the bacteria to grow everywhere. And we call that a lawn of bacteria, where the grass in a lawn is growing everywhere in the lawn. That's why it's called a lawn of bacteria, by the way. And then you put the cotton swab in the biocide container for cotton swabs. And then you put your lid on the plate. And you let that sit for 5 minutes. And then you add the disc for antibiotics at between 5 minutes and 15 minutes after you've added the bacteria. You give it 5 minutes to let the liquid from the bacteria to be absorbed by the plate, the agar plate. You appropriately label the plate, and then you incubate the plate upside down or agar side up. Let me get this out of here. So this is the normal plate. You then incubate it upside down in the plate for 35 degrees. And we always use overnight, but as you know, the exact procedure is something like 16 to 18 hours for the Gerby-Bayer method, which isn't quite practical in a microbiology lab because it has to be 24 hours. But we incubate overnight, and then we look at the plates and measure the zone of inhibition. Any question about any of that? I once had a student who, when she inoculated the plate, simply went and then she had a whole areas of the plate which were not swabbed by bacteria. And then when she put her antibiotic disc down, she couldn't measure the zone of inhibition because there were all areas of her plate. And I told her, well, you didn't swab the plate everywhere with bacteria. And so she had to either do the experiment again or simply discard the plates in the biohazard container and then use her lab partner's results. So make sure you swab the plate everywhere. All right, so then watch the video clips for preparing a culture for Kirby-Bayer disc diffusion. This is following a standard technique so that we reduce the variables. And the main thing is to make sure the bacteria are growing for a certain amount of time and not longer than a certain amount of time. And then you have the bacteria growing to a certain depth, and it's something like 0.5 McFarland units or 1 times 10 to the 5th cells per millimeter. Make sure that's right. Yeah, 0.5 McFarland units, 1 times 10 to the 5th cells per mil. But there are some other things. Where was I? And then watch the video on interpreting the results for the Kirby-Bayer disc diffusion test. You need to see that because you're going to be interpreting the results of the Kirby-Bayer test. And then the virtual lab. This may take a little while to open. I've got a fairly fast internet connection, so it doesn't take me very long. And then open this, and you can read the description, the steps, the credits, and the start. We're just going to look at the start here. And you can see the Kirby-Bayer disc diffusion susceptible test here for E. coli using these antibiotics. Here's the key here. So E there is, I believe, erythromycin. Yep. No zone of inhibition. TM, I don't know that one. TE, where is that? TE tetracycline, right there. What you can do is take the ruler here and measure the zone of inhibition across the diameter. And you can really only do it one time here because you can't make the ruler move. But we have this one, 17 to 18 millimeters for TE, which is this one. And if you get it right, it'll be green. If you get it wrong, it'll give you a red X. Oops, I didn't do that right. So TE is there. Sorry, I wasn't looking at TE. So 17, that's actually intermediate, which is correct. And this is for E. coli with these antibiotics. And you can hit the menu button and restart again. It'll switch the plate, and it says Pseudomonas aeruginosa. And for C right here, Zone of inhibition is about 17 to 18. Ooh, let me measure that more carefully. 17 and 18 are actually different. I switch my glasses here, I get the more powerful ones. All right, that's about 19, almost 20. So that means it's oh, sorry, that's 15. That's 14 or 15. Sorry, I misread that. So that's here. Okay, that's how you work this. Hit the menu again. There's a third plate, and this is Staphylococcus aureus for the different antibiotics. And this looks like it's actually MRSA because only vancomycin is showing a zone of inhibition. All right, any questions about that? You will need to do this for answering some of the questions in the lab. Two, using the standards required for the Kirby biosusceptibility test, an E. coli culture is prepared using the spread plate technique. Antibiotic discs are placed on the surface, incubated overnight at 35 degrees, and below are the results with the NCCLS standards for interpretation. So here we have the, let me shrink that down just a little. Here we have the zone of inhibition for ampicillin, and we're measuring it with calipers. We're only looking at E. coli, is that right? E. coli. And then we have the table for the NCCLS standards. So the first one I'll do for you, ampicillin, right here, that is not measured from here to there. It's actually measured from this 0 here to that 0 there. So that's just a tad less than 15. We'll call it 15 millimeters. Which ampicillin means it's I, intermediate. So you write that down here, 15 millimeters, ampicillin. The interpretation, it is I. And then do that for SF, GM, and TE. SF, GM, TE. Get the measurement, compare it to the standards right here, and then tell me if it's R, I, or S. Resistant, intermediate, or sensitive. And then fill in this table. And I'll grade this table. Any questions about any of that? All right, let's go to the worksheet now because I'm actually on the worksheet. Only submit the worksheet if everyone were to turn in the lab module, which most of you cannot do because I think I've removed the questions from the lab module. It would quickly fill up my Canvas web page, and then you guys would not be able to submit an assignment. So answer 2 here for this question, fill in the table. And 3, list 5 variables that must be standardized when performing an antibiotic susceptibility test using the Kirby-Bayer disk diffusion method. And there's a table of them in the lab module. 4, the size of the zone of inhibition often varies for 2 different species of bacteria when challenged with the same antibiotic. What is a possible explanation for these differences? Give that there. 5, what would happen if we didn't use aseptic technique to put antibiotic disc on the plate? Answer that, and then mention what will happen to our test results if we didn't use aseptic technique to put antibiotic disc on the plate. What would happen to our test results? 6. What would happen if we increase the concentration of the antibiotic in the disc? Be specific in your answer. And then what would happen if we decrease the concentration of antibiotic in the disc? Be specific in your answer. 7. Antibiotics can be bactericidal, they kill the microbe, or bacteriostatic, they inhibit microbial growth, but do not kill the cells. When observing a zone of inhibition around a disc, how would you determine whether the antibiotic causing that zone was bactericidal or bacteriostatic? I'll warn you, this question is a thinking question, and students frequently get it wrong the first time they submit their answer. Unfortunately, most students do get it right the second time they submit their answer. So this is a hard question. 8. When performing antibiotic sensitivity testing, it's important that the test conditions are kept very standardized, meaning we always use the same kind of media, MH media, always have the same agar depth, 4 millimeters, always the same age of the culture, a culture that's been growing for, well, less than a day. Why do we do this? 9. Are there other substances besides antibiotics we could evaluate using this method? By this method, they mean the Kirby-Bayer disc diffusion method. Oh, and I forgot to mention something. Let me go and talk about that. The Kirby-Bayer disc diffusion method is very good at telling us which antibiotic to use for a given bacteria patient, because the antibiotic diffusing through the agar is similar to the antibiotic diffusing through a person's tissue and then fighting off the bacteria. And so the Kirby-Bayer disc diffusion method is very good at determining which bacteria or which antibiotic the patient's bacterial infection will be sensitive to. With that stated, usually the clinician will not perform a Kirby-Bayer disc diffusion method on a bacterial infection. Usually they'll assign the antibiotic that is routinely assigned for what they think the bacterial infection is. Like if they think it's Staphylococcus aureus, they will assign whatever is commonly used with Staphylococcus aureus. Some of you guys may know that. It might be carboxycillin. I don't know, something like that. It's no longer penicillin G, but something like carboxycillin is the standard for using for Staphylococcus aureus. But once that doesn't work, they will then fall back on the second antibiotic, and they may do the Kirby-Bayer test at that point. Or if the second bacteria does not work, they then will do the Kirby-Bayer test to find an antibiotic that will work against that bacteria that's causing the infection. Now, there's a lot of things they have to know here, and that is what bacteria species is causing the infection. And of course, they already know which is the routine bacteria to use against that bacterial infection. So they don't usually start with the Kirby-Bayer disc diffusion method, but they use it if the standard antibiotic does not work against that bacteria. I guess we're not here, so let's go back to the worksheet. All right. I think I talked about 9, so let's talk about 10. Does the presence of a zone of inhibition necessarily mean the bacteria being tested is sensitive to that antibiotic? Explain your answer. 11. From the size of the zone of inhibition of chloramphenicol on E. coli in the virtual lab, is the bacteria sensitive, intermediate, or resistant to that antibiotic? For chloramphenicol on Pseudomonas aeruginosa, is the bacteria sensitive, intermediate, or resistant to that antibiotic? All right, any questions? If not, I will be here till 8 o'clock to answer any questions. Some of you can use my help, should use my help, but that's not required. I know a lot of you are getting together and sharing answers, and if you do that, you should make sure that the answer you're using is the correct answer, because I'm finding a lot of students who are getting the same wrong answer. All right. I will be here until 8 o'clock to answer any questions. I will be here until 8 o'clock or the last student logs out.